This afternoon, we have 3 breakout sessions. One of the things that we've been trying to do over the years is to show people the diversity of applications of CompuCell3D. And we're always very grateful when people are willing to come and talk about their projects they've implemented with CC3D. Today, we have Chandrashekar Kuyyamudi from Harvard, who's going to be talking about heterotypic intercellular adhesion to its efficiency of cell-on-cell migration. We're very grateful to him for coming to talk to us. As usual, I need to remind you that this is recorded for future viewing. And he's also put some demo code in the chat that you can download to run the code that he's going to be showing today. And as always, I'll be available to take questions in the chat. And I don't want to take any more time from him, so I will turn the screen share over to him.
Thank you, James, for inviting. And before I start, let me thank the developers, the creators of CompuCell3D software, and the members of the organizing committee who have been doing this for a while now. And thanks to all of you for taking an interest in this and attending my talk. So this will be about what went into simulating something relevant for our work that got recently published. But this is an interesting project to present to people because this also happened to be the first project where I used CompuCell3D. The title of the talk is Modeling Germ-cell Transepithelial Migration in CompuCell3D. I will just share a little bit of the biology that is relevant here. Won't get into too much detail or the biological results from the paper because I've been told, and it makes complete sense, that that's not the focus of the workshop. So if one were to look at the early Drosophila embryo from the point of view of what we will refer to as the germ cells or the pole cells, which I might interchangeably use today. From the dorsal view of things, they are formed at the posterior tip of the embryo, about 2.5 hours after the embryo has been laid by the mother. And during gastrulation, they end up in this epithelial sac called the primordial-midgut. And then they individualize and exit the midgut and form bilateral clusters that will migrate towards the gonad. And that's where they basically meet somatic gonadal cells. So together with germ cells and the somatic gonadal cells, they form what is referred to as the embryonic gonad. And then later on, the embryonic gonad during the larval stage becomes a larval ovariole or the larval ovary or the larval testes, and eventually in the adult, they become the gonads. But for us, the most important part today, the relevant bit for today is when the germ cells during gastrulation end up inside this epithelial sac. How do they manage to exit? So because it's a solid epithelial, it's a single layer epithelial barrier that they need to breach in order for them to exit. And only then they form their bilateral cluster and they migrate towards the gonad. So this is what I mean. This is another schematic showing the same thing. And here's more of an actual video of this process. Again, looking from the top of from the dorsal side, we see these germ cells exiting the barrier. And our interest in modeling this, again, another view, just this is a lateral view of the same process. What we know and what we knew for a very long time is that the cells at the early stage, once they're in the midgut, they are in a big blob, which is they're all bound to each other by E-cadherin. But when they exit as individual cells. So they individualize, and there is a lot of previous work showing that they downregulate their E-cadherin on the surface, uh, which allows them to basically become individual cells. And when they exit as individual cells. So that is something that is very key. Just in case, if I assume E-cadherin here is just the addition molecule that is on the surface that promotes addition between uh, different cells who also express E-cadherin. So that is what we are talking about. So for most of this talk, I don't think one needs to know anything more about the E-cadherin. All right. So as you can see, when they have a lot of E-cadherin, since all the germ cells do have that, they have a tendency to bind to each other and form the blob. And for them to exit, they need to separate from each other, individualize. And previous work showed that they do downregulate the E-cadherin so that they can become individual cells. But it's also true, previously and also from our stains, that it's very clear that EA was as a marker for the germ cells. But the key thing is to look at the E-cadherin channel, which is shown, just the E-cadherin channel is shown below. If you look on the surface of the edges of the germ cells, they still have E-cadherin, even though they have downregulated and basically become individual cells prior to migration. They do have it, and they do have that E-cadherin on the surface while going through through the epithelial barrier. And we wondered, does it play any role? Because there are also experiments where when this was perturbed, things were either delayed, or when there was too much of it, they failed to basically exit. So given these things were not very clear, we wanted to know whether there was some sort of an optimal um, E-cadherin levels on the surface of the cells that allows them to separate from each other, but plays aids them to exit the midgut, which is an epithelial barrier. And the epithelial cells, one thing to know about them is that they are confluent cells and they express a lot of E-cadherin. In fact, E in E-cadherin comes from the epithelium. Um, so naturally, they allow of E-cadherin, and germ cell E-cadherin levels are significantly lower compared to the epithelial cells, but nonetheless, they do express E-cadherin. So this was our question, and the answer turned out to be, at least from our experiments and so on, which is that it is yes. And people who want to know more about that, more about this, can read our paper. But today's talk is about how I went about modeling this process using CompuCell3D, and that's what we will focus on. Okay. The first thing is we wanted to take this 3D epithelial sac and reduce it to 2D. So if I take a slice of it, I will get something like an epithelial ring. So that's the first step, which you reduce this three-dimensional problem to two-dimensional problem. You want to make an epithelial ring. And that was the first step. There are a few things to consider. Given that they express this E-cadherin, which means they have affinity to bind to each other. And if you run just a simulation of a few cells, bunch of epithelial cells which have E-cadherin, enabling them to sort of adhere to each other, you will get a blob of cells because that is the energy minimizing surface, because it minimizes the surface and not just the surface and the overall surface area of the whole system. It also minimizes the contact with more from the CompuCell3D. From the cellular pulse framework, they have the least amount of energy, interaction energy, when they bind to their own kind, and that is enabled when they all form this solid clump rather than form a ring, because ring leaves a lot of surface which is not bound to their own kind, but rather it could be some other cell type where the interaction costs are much higher. Or in other words, the epithelial ring is not the energy minimizing state. So this has to be sort of dealt with, because we are dealing in two-dimensional, and it's important we have this system so that we can study what we want to study. So one way was to add and define the lumen in the ring itself as a cell type. what it is, for some historical reasons which I can't trace back to, I called it the cell type W in the code. And then that, in a way, prevents cells from collapsing. But to also to make sure that the epithelial cells don't form, in some case, you don't want them to deform when a cell is trying to go through them in such a way that they basically form like not just a single layer, but rather multiple layers or any serious deformation, which seems very unrealistic. A good measure, we assume that to have the kind of elastic properties of the epithelial ring, epithelial barrier, we added adjacent cells to be sort of connected by springs. And this is something that one can do in CompuCell3D using the FocalPointPlasticity plugin. So that's what we did. And before I before I get into this, I want to say that this was the first CompuCell3D project that I did. Prior to this, I had not done any work in cell parts model for that matter. Only during my postdoc, I decided to sort of work on CompuCell3D, and the first thing I did was to reproduce some of the results with my own codes for the version paper by James, the Grenier Glacier model. And later on, when I wanted to study this problem, I used to write my own code in Julia for simulating this process. And I want to highlight that in the past, I used to do it a bit differently, which is before CompuCell3D. The way I used to do this was I used to assume an annulus. An annulus is like a halo, a region in the in the medium. Only the epithelial cells had low interaction energy, where the interaction energy would be very high if they left that annulus. So this, in a way, was basically enforcing them to maintain the ring shape, still allowing them for some movement, but not so much that the epithelial ring, structure is destroyed. So this is the way I used to do this before I started to switch to CompuCell3D. And suggestions about having a cell in the middle, basically the lumen itself, modeling lumen itself as a cell, and adding the springs between the cells was something that um, that I learned, and James advised me when we had a couple of calls on this. This is what he suggested, and which in the CompuCell3D in the CompuCell3D context was the very sensible thing to implement, and it was very straightforward to implement. So I'm sure I'm told that I think focal point plasticity is something that you will learn in the next week, maybe if you haven't already. So this allows you to sort of, if you have different cell types, in general, you can basically, for every pair of cell types, you can define this. The key parameters here are lambda, which in a way can be seen as some sort of spring constant. Activation energy, the target distance, which is the resting length of the spring, and the max distance. The max distance is like if there are other forces that basically make the cells go further than this, the springs just break. So here in this case, we only wanted this to be between two things. One is between epithelial and epithelial cells. So the adjacent cells have this. And we also had one for good measure from the lumen to every epithelial cell. So, or in other words, this is how, if you use in your CompuCell3D visualization, if you allow for focal point plasticity links, FEP links to be visible, this is what it will look like. Usually when I show the simulation, I usually disable it because it kind of makes it, let's say ugly, but this is at the heart of it, this is what is happening. This is how the epithelial barrier is being sort of the ring, and how we ensure all what we want from the ring. So just as a minor digression, it's an interesting thing to think about. What is the minimum constraints one needs if you want this group, bunch of epithelial cells to form a ring instead of a blob? Of course, here I'm using something to sort of enforce this shape, allowing only the kind of freedom that I wanted. But in general, like if you have a bunch of cells, how can you sort of make the state where they form an epithelial ring the energy minimizing state? Under what kind of constraints can you achieve this? Or in another case, in 3D, this would be like basically forming a shell of cells rather than a solid spherical mass. And this is something in developmental biology, people must have heard of this term called the blastolite during the early development, where sorry. So this is something that is a particular state that the embryo achieves during development. So again, I just wanted at this point want to thank James for and this cat for taking the time to Zoom with me and basically help me basically switch to CC3D, which then on for any CPM simulations I've always used CC3D. All right, this is something that I already mentioned. This is how I used to do before CC3D. This way of so for example, to implement this exact thing in the CompuCell3D probably would be much harder than just simply doing what I did by having a define lumen itself as one cell and having the FPP links. All right. The next step is, of course, you want to have a germ cell because you are studying germ cell. How the germ cells or a cell basically goes through this epithelial barrier. So this you define another cell, which I think in the code is the cell type G, and then you wanted a radially outward chemical field. There are ways to probably like code this to have this field, but I just wanted to make so because I didn't in my simulation I was not interested in basically making the field itself dynamic, but rather this is a static field that exists. So I took the option of using if you go to the flexible diffusion solver for your fields, the code, there's a place where you can just basically give the path to a concentration file. The file basically with XY for every XYZ pixel or voxel, it assigns a chemical concentration. And that's a file I generated and I just basically fed it because it did the job because I didn't want to do anything with it. And of course, after to make sure it remains static, one thing you want to make sure is that you set the decay constant and the diffusion constant to 0. So that it's just a static field. And I left a bunch of lines below it, which you can totally delete and nothing will make a difference, the boundary conditions and so on, because this is a static field. Since there is no diffusion or decay, there's no dynamics of this field. And then the chemotaxis part was pretty straightforward, which is that you use the plugin Chemotaxis and you basically say what cell type is chemotaxis, type G, and you say you also define lambda, which is a strength. Any positive value is basically like it's attracted to it. I think you can use a negative value to basically kind of move away from it. And in this case, you have to specify which field is it, sort of chemotaxic. In the beginning, when I used to write my own codes, as well as when I used the CompuCell3D when I switched to CompuCell3D, for a long time, I used to still basically work with the energy matrix or the J matrix, where you specify the pairwise interaction energy for different cell types. That's what I used to use. But then I realized since one can actually, you can actually make the instead of using interaction energy as a proxy for whatever the adhesion energy because of the adhesion molecule, you can just directly use the adhesion molecule or density of these molecules on each of the cell types and basically define a function that tells you what is the addition strength or the energy as a function of concentration of the addition molecules on the two surface. So here, as you can see in the XML file, you can just define this molecule. I call it E-cadherin, E-cad. On the lumen cell, I set it to the density to 0. The epithelial cell, I set some non-zero value, and which is large, and in the germ cell, I also set it to a particular value. Yeah. This is my default values here, but in the Python steppable, I do change it. I know later, but this is how you define this. And then the most important thing is you also define something called a binding formula, which is how does the interaction energy in some sense is a function of these molecules and the density of molecules on the two cells. So one of the options, I think this might be the default option there, which is minimum of the two concentrations. Okay, under what conditions is this a good function? It says the implicit assumption about the binding here is, of course, the binding energy, the lower, the more number of addition, the binding is stronger, and the interaction energy is lower when there are more addition junctions that are formed. And the number of addition junctions that are formed is just, so we are assuming that those junctions are formed very rapidly, and almost all of them are involved in forming the junction. And so the maximum number of junctions you can imagine two cells with number of molecules A and number of molecules B, the maximum number of junctions they can form is whatever is the minimum of A and B, because that is a formula that I set here. I haven't played around with like using different formulas, but I think at least for our purposes, they did pretty well. But it is important because I'm sure that there are other processes some of you want to model where you want this to be a bit more nuanced, but this is what we chose. So just to show the simulation, this is how a sample run would look like. A germ cell that starts in the middle and just sort of, goes through the barrier. And I basically instruct in the code after it, goes beyond a particular radial distance from the center for the simulation to stop. I note down, and in the code, I just note down the time, the position of the cell as a function of time. All right, this is the time to show the first demo. How do I switch? Sorry about this. I think I need to. Let me show you the code. Hopefully you can see this. All right. So running through just the XML file. Yeah, okay. Okay, well, I set a temperature here. I did change for my study, I did change different temperatures, but I didn't report it much. And it's an easy parameter to vary here. I hope everyone can see the code. Again, the four cell types, which is the medium. Then you have the lumen, the big cell in the lumen is the W cell type, and E is the epithelial cell type, and G is the germ cell type. In this case, I just hard coded the value of their original sizes, but you don't have to do that. And again, the surface. And for the cell W, which is the lumen, I didn't have a surface constraint. So because the whole objective of having that was to sort of prevent the whole ring from collapsing. So that didn't have a surface constraint. So I did keep all of this. Did I make good use of it? Probably. I mean, I did use it because I think the center of just by calling the CenterOfMass plugin, I get to easily access the later on in the Python step of like, I can easily access the center of the cell, which is what I use to know where it is in the whole with respect to the ring as a function of time. I did also keep for good measure NeighborTracker, which is useful because if you want to know when is the point at which it first made contact with an epithelial cell, it helps to basically have a quick way to access who's in the neighborhood of a particular the migrating cell, and you can quickly ask like, okay, if it has an epithelial cell in its neighborhood, which means it has made contact now, and it by just calling these plugins, it makes it very easy to access this information if you need it. Just highlighting you like how easy it is to sort of define these things. Usually, if you don't have AdhesionFlex, which is where you explicitly model the molecular concentration, you would have something called adhesion, I think. But here, as I said, as I showed before, this is the part, and this is the Chemotaxis part, and this is this is massively underutilized flexible diffusion solver plugins. There's almost nothing for me here because I'm just hard coded. I have a static field and I've set the decay and diffusion coefficient to 0. This is the focal point plasticity. If the moment you just call this function, you add this plugin by going, I mean, I hope you're not all familiar now. Sorry, I think if you go to plugin here and if you click on this, which I'm not going to do because I could delete few things later. Focal point plasticity. By default, it lists this thing for every possible pair in your simulation. In my case, as I said, the only thing that was relevant was between epithelial and epithelial cell and the lumen-cell and the epithelial cell. So I just basically delete everything and keep what I want. And the initial state was defined by this PIF file where you just basically say for each what cell number, what cell type occupies a particular voxel in this thing. So that's what I used to use. And then you have the steppable file. Forget about saving the cell field and everything. Those are things that I wrote to sort of, access the, if I want for any visualization purposes, if I want the chemical field or something, or the cell, how the state look like, that's what I used to do. But here's the important thing, which is that I used to set the lambda, which is the strength of the chemotactic field on the epithelial cell here. So it was easy, so that later on it was very easy to sort of. So I think I can do this. No, yeah. So you can just basically select this and say add to scan. This is something that is useful, and I think if Hayden told me this is something that you learn probably in the next week. When you want to do a parameter sweep, it is as simple as this. You define a parameter exploration, and once you're done, all you have to do is to add to scan, and this goes into that, and you can define the range over which you want to scan this parameter. It's as straightforward as that. So that's what I used to do. This was on a parameter scan, and the parameter obviously was the amount of E-cadherin on the epithelial cell and the amount on the germ cell. These are the three key parameters that I changed in the thing. And I also changed the random seed because I wanted to do like 100 or 500 or 1,000 independent iterations for every parameter set. So it was easy. By keeping the random seed itself, a parameter that I changed for 100 iterations, I just keep changing the random seed. So that's what I did. And the rest of the code is pretty straightforward, which is you want to basically define. So what I do is I pick a random angle at which I want to seed a germ cell in the beginning. And then I set the target volume based on target volume and I basically also set the chemical lambda to whatever that I was defined here. And again, this is where I set the epithelial concentration and E-cadherin concentration of the epithelium of the germ cell. So whatever value I had here is sort of here is pointless because it's kind of overwritten by what was there in the steppable file later. Each step, all I do is to basically get the position because I define the center of mass. I can easily access the X coordinate and the Y coordinate of the cell, and I can basically ask if it's in contact with any, any neighbor, which is epithelial cell and so on, and I ask, has it crossed this distance from the center, which is 70. In this case, I set as 70. And if that is the case, say, stop the simulation, because why waste time? And you also need to, because not in my simulation, not all the time they actually cross the barrier. So you also want to set a maximum time, or other words, it will go back to whatever is the maximum time set here in the XML file, and it will run till then. And I had to basically say if it ran out of time, then basically then there's a function called the finish that is called, and you can say, in both the cases. So the finish function will be called, and if you satisfy any constraint and any sort of criteria, in this case, which is to cross the distance 70, if it stops simulation, then it goes to this function, which is on stop. In both the cases, I just stored, I stored them as a data frame. I just that's the beauty of it, which is that you can call every standard Python library from here, which in this case I use pandas to basically just store this whole thing as a data frame, so I can just write the whole value in as a CSV file. That's what I did. I'm sure there are clever, better, probably inbuilt system to save all this information without having to call all this, but this is what I did. All right. So what does the simulation look like? Okay. So this is with the FEP links being shown. I can just, maybe the first one you can see with, with all the FEP links visible. That is it. So of course, I don't know why this changed, because I think I did something with the colors here. All right, this is without it. We can talk about this in detail more, but let's return to this presentation. So in the initial code, as I said, I used to use something called a PIF file, which is not needed in this case. But there are other simulations of CompuCell3D simulations where this is very useful, especially when you want to have an initial configuration that is from a, let's say, micrograph of bunch of cell types which you're stained for, which I do use it. Maybe if I find time, I can just use that file and show you what another project of mine where I do use that. Is where it is the best tool to do it, which is to go segment it, get the values, get the pixel values. Based on the actual micrograph, you can basically assign them different cell types and cell numbers, and then import that. So transfer the whole thing into a PIF file and then import it. So the initial configuration could be something that is, true and very faithful to an initial state that you're seeing in a biological system. In this case, it's artificial, and there was no need for this, but this is what I did. So if you see the columns here, it would be the cell number, cell type, I think, followed by the X. Oh, sorry, it's just the bounds. It is 57 to, because you can actually, if you want to define a rectangle, for example, I can just say 57 to 60, and from a y coordinate go from 60 to 65. I can do that. In this case, this is every pixel was basically, it's every individual pixel was coded. So it was 57 to 57, 60 to 60, and z coordinate was 0 to 0. So this is, yeah, it's now funny when I think about it. But that's what it did. But doesn't matter. The code loads it once. Doesn't matter. It almost is not about it. Not going to affect your performance at all, because much of the time is consumed in the code in running the simulation. It's very ugly, but it works. And in fact, this is the best way to do things in a different other context. Which I can show you another one where I think this is the way to go. So just to show um, the code, I will just close this. Thanks for my very extremely bad naming. I just changed the folder name, but it's the same code. But here I've made it different. For example, you can create an epithelial. You can define a function in the stepable where I say create an epithelial ring, and I mean these codes are available to you, so you can take a look at it because it's very easy. Once you create an annulus, all you need to do is to basically go if there your whole annulus is 0 to 360 degrees, and if you want to 20 cells, you basically assign a certain theta 1 to theta 2. So whatever is the angle range you want to assign to each of the epithelial cell, and you basically give them different cell name and often assign them cell type. That they can do. You don't have to use a PIF file for this. And this is how you basically every time you define a cell, you can now say whatever is the volume of the cell, current volume, set it as a target volume, and you can also set a lambda for the volume, and that's how we do things. And I also told you in the chemical field, I just assigned the chemical value at XYZ, every XYZ value. The chemical field here was very straightforward. It's a radial field. So you start from the center, you assign whatever is the radial distance as proportional to the chemical field to be just proportional to whatever is the radial distance. And I just normalized by the largest value. That was just a simple field. So I just that's all I did there, which is again, few lines of code in your stepable itself. You didn't have to use a text file for this or anything of that sort. You could just simply do this here. Just as a proof of principle, you'll realize that in the flexible diffusion solver, you don't need these things anymore because all of that is done here and it will work identical. The boundary conditions, all of those things are not important here. I mean, I wish I had a good example of mine where I've made very good use of this whole boundary conditions and so on. Unfortunately, so far, most of the time I've been working with pretty simple fields. But I'm sure you'll see things where this has been put to good use. Chemotaxis, I can plug in the same thing, nothing else has changed, except just to show that, you can use them. While I'm here, I might as well show you where I think, yes, PIF file can be very useful. So I think in this case, this is another project of mine where I do use a PIF file here, I think. Okay, so here here I import from an actual file. So for example, this is what I mean. Yes. This is a case. This is an actual tissue where there are these bunch of different cell types, and I wanted to sort of start with the initial configuration. So here, this was a case where I just load them from actual data, and then start the simulation from that point onwards. So in these kind of cases where you want to you're not creating some simple geometrical configuration, but rather want to take it directly from a biological example, PIF files can be very useful. Going back, just to show this the simulation run here without the PIF file or anything. Oh, this is actually a good point. By accidentally, I left it there, I think, but it highlights something useful, which is that right now, instead of showing the cell field, it is showing the chemical field. But you can always switch to chemical field. Here it's more clear what I mean by the radial field. You can look the cell basically live there, and you can see nothing changes in the field. That's because it's a very static field that I've considered here. All right. Which I showed you already. All right. I was told that it's good to sort of leave few instances of how you can extend. Sorry. How do I get rid of the? Anyway, I can't get rid of the floating panels, but that's all right. So one thing that might be useful to do, as especially not because I think this problem itself should be explored in greater detail or anything, but I think as an exercise in CompuCell3D and mastering some of the tools that it has, and which are definitely not covered in my simulation, which is a very powerful tool to use multi-compartment cells. And I'm sure there are some speakers or like you will learn about this, which is right now each cell is a single compartment. It doesn't even have a nuclei, even though all my schematics have a nuclei. I don't even define a nuclei there, but you can have a nuclei as a different compartment. But especially for epithelial cells, we know that the epithelial cells don't have the same E-cadherin distribution throughout the surface. So they have something called the apical and the basal side, and it is useful to have that polarization of epithelial cell for certain context. In this case, if you want to make these epithelial rings to be not of single compartment, but to have these both apical and basal side, one straightforward way to extend this to have these two sides. One way to do this is to basically define each cell as a two compartment cell and assign different properties to the two different compartments. Another one can argue that it probably is not two, it should be three. It's like an apical, basal, and whatever is in the middle. The bulk should have different properties. So you can extend this. And I think that especially when you break this cell into two, I think you'll realize that you have more and more compartments. You want to take a look at it and see if the cell looks realistic enough to you. And are there any tweaks and so on to basically make it look more realistic, or what you want, as close to the actual biological phenomena you're trying to model. Another straightforward way to basically straightforward thing to include is, I think, nuclei, especially nuclei, is pretty easy to do. It's just defining compartment which is inside. One way to make sure it stays inside is you make it it has very low interaction energy with the cytoplasm. You make it such that it stays inside and make it very high, very costly for it to basically leave the cell by making very high interaction energy for anything that is outside the cytoplasm. It's an useful thing to have because in many, especially cell migration cases, we do know that the nuclei have different properties to the cytoplasm. It's not as elastic. So the narrowest gap you can go through sometimes is dictated by how distendable your nuclei is. So it would be nice to, especially if you're modeling cells cell migration and so on, it would be nice to have explicitly the nuclei with different different elastic properties. Or like in this case, what I mean by in CompuCell3D language, that would be like stronger lambda surface. Which will allow you to sort of explore the consequences of having a more rigid nuclei and what it does to basic cell migration and so on. And you can have the same thing for the epithelial cells as well. It's probably a first nice exercise to do once you learn about multi-compartment cells. Another thing is, of course, in this specific problem, you can actually, I did do this at some point. I used to have 4 cells, and I realized that if biologically there are like 40 close to 40 cells, and if the cells are living individually, like if I had to work with some number, I think the one is a good start. One cell is the best state to start from. So I stuck to it. But of course, there are interesting things you can study because in the actual process, the multiple cells are trying to cross the barrier at different parts, and this itself might actually affect each other's ability to go through elsewhere, because in a way their ability to go through could be like affected by some other cells trying to get through in the other parts of the barrier. So, which is not unrealistic to think of. So, another natural way to do is to have multiple cells and see what it does to the overall simulation. Okay, I want to open up for like a lot of questions. Before that, I want to say a few things, which is to conclude. This thing I want to say, which is a lot of us have a lot of hunches, ideas, and hypotheses on a regular basis, and we want to be able to test them quickly. And CompuCell3D is an extraordinarily useful tool when something can be tested in a CPM framework. I know the amount of time is to spend before I test a few things, before I used to write my own code. Like, however trivial it might be, there is a significant amount of times that goes into basically writing that code. And every time you write this code, you also, for every different setting, you need to basically simulate you have to visualize it to basically build an intuition and see what is wrong and whatever, to test. Like, visualization is the best way to sort of check if things are happening the way you thought it would be happening, or like, is it a useful model and so on. The fact that visualization comes for free here is one of the strongest thing about CompuCell3D. And most ideas, at least in my case, are not worth pursuing further. And it's very good. The CC3D helps you to attain closure very quickly. You can do this and you quickly realize, okay, you're wrong, or maybe it's stupid, and you move on. So I think that this ability to sort of basically quickly, attain closure is an extremely useful tool with a useful factor, so that you can move on to basically new things and new hunches and hypotheses and whatnot. So I think it cuts down the whole time that goes into basically testing your hypothesis. Um, Apart from all the other useful things that it has. So this is, for me personally, these are some of the great things, and this is why I continue to use CompuCell3D every time I have something that can be um, framed in the cellular parts framework. I also think that I think you will also learn that it doesn't just have to be CompuCell, a cellular parts framework anymore. I think the CompuCell3D tool itself, the package itself, has become quite general, that you can do a lot of things where um, uh, it's a good place to simulate. Like, even if you have a single cell just migrating in an uh, chemical field, I guess this is a good place to start, even though it probably is not doing anything. Uh, like, you're not really utilizing the cellular parts framework there so much, but I think it just has gone beyond just the cellular parts framework. I think this is my feeling. Yeah, so I would like to thank the people who funded, especially in the early days where I was working on this particular project. My collaborator Shorit, who did all the experiments, and my PI Cassandra Extavour. Thanks, members of the lab who are great colleagues, and it's a nice environment to do work. So again, I want to thank the organizers, and I'm more than happy to answer any questions anyone has. Thank you.
Thank you. Any questions in the chat? Did you want to show the code running, maybe?
Yeah.
You have it on the desktop. Maybe other people. Could people try to follow along and download and run the code that he provided? I was able to get it to work. Were other people able to run it?
I think the seed is set to the same value, so I think it always runs on the same place, but that's something that I can fix. Yeah, maybe this will make the cell go in the other way, some other direction. Hopefully, yeah, there you go. So, and something that we learned, of course, is when this has like when the adhesion is quite strong, which I can simply tweak by making the germ cell epithelial constant density go maybe high. I've been told from James and many others to always do save all, which I think you will hear in the coming days. uh,
So maybe I can
that's
ask
it.
a question, which is,
Yes.
when you were getting started, what was the most difficult thing to design the model? And then after you decided what you were going to model, what were the most difficult steps? We talked a little bit about that, but what were the biggest obstacles in getting going on this problem?
Um, I did take a while to sort of the very fact that I was uh, simulating. I had to maintain a ring of epithelia, which is not the natural, not the energy minimizing state for a group of cells that have addition affinity for each other. Um, means that I had to have additional constraint to ensure that I do get the ring. And that always, whenever you make such things, it always bothers you because you know how artificial are you making things. And it's not like I've had I'm like today happy. Of course, these things will always be there. I think it's uh, it's something that only with time and playing around you'll realize. So I definitely felt a lot more comfortable with having something like a lumen, which is a cell, because we know that in the blastula, for example, the fluid pressure makes a big contributes to basically maintaining the shell of epithelial cells, for example. So this is something more akin to that. So whenever I think if you're doing something artificial, like the closest it gets to something that is biologically meaningful in some way or the other, you feel that much more. I would say that less discomfort when you do this. I think that will always be there when you make too many assumptions about certain things. So the ring, I think, to finalize how will I get this was basically it took me a while to sort of keep thinking about adding the springs between them. How realistic is it? What is it actually doing to the thing? Running a few simulations without it and seeing what it does and trying to keep it the bare minimum. Um, Another possibility is that when in doubt, maybe just take that as a tunable parameter and run simulations under different parameters and see what it does to the simulations, because that itself probably is some parameter non-control, sort of in a way regulates the epithelial property, epithelial ring property. So those are the things. So a lot of these parameters which are never touched in the last few years were were explored thoroughly in the beginning. Not thoroughly, my bad. What were at least explored in many different ranges, and finally when it looked okay, I stuck with those things and just focused on the parameters I wanted to explore. And I think this is again to point out that this is why I think right away when you write your code and you have the visualization tool, it helps immensely because I think otherwise for everything you want to, the fact that you can see the simulation then and there helps a lot to basically tune it very fast. And I think that especially that first few days after you have written something to tuning these things, I think the CompuCell3D visualization tools are super useful. And it's fast. It's really fast. Um, I think as my default, I think for most people, the default is what it updates every uh, 10, 10 steps. You can make it faster, slower. So I think this is very, very handy tool. And of course, in some cases, I do disable it because it takes very long to run. But I think in these kind of things, it was easy. Uh, and in some cases, I have uh, scaled on the whole model and played around with a much smaller system, and then later scaled up every cell and everything later on. So I've done that later on. Um, um, so yeah, so I think one of the things that was also a bit tricky was um, so um, documentation thing, which was I think now it's probably easier. Uh, uh, so I especially every time I use a new new feature in the CompuCell3D, I just write it down in some document of mine because I realized that, half the time in the beginning, it used to be like, if I don't use that feature for long enough, I just forget it. And then I know that, but it is at least nice because now I know for a fact it can be done. It just, I don't know how to do it. So I think I kept track of these things. I always refer to the projects where I basically use these things. I think I'm sure that there are more and more examples and more tools. That was the easy way out. And I still think Claude, ChatGPT, and all probably just makes up commands that don't exist in CompuCell3D. And if people have played around with it, they'll realize it will just simply say some plugin which doesn't exist. It sounds very legit, but it doesn't exist at all. So those problems are still there. So I think it's every time I learn something that I can be done in CompuCell3D, I store it so that I can go back and refer to it. So I think that yeah, it's yeah, but um, I mean, of course, meeting you and discussing how to go about this made a big difference. I must say that writing the own code at some point in time did help. I mean, I think it did help me. I think it was very nice to know the underlying this thing and do this. And something that I'm very glad I don't have to do it. I remember when I wrote my initial code, one of the most annoying things to do was to constantly check for connectivity of cells. And you realize that much of your CompuCell3D code is basically that particular part. If the code is not very sophisticated, but still you have a big chunk where you're basically finding an efficient way to check if your cell is connected. So you write your breadth first search or whatever, depth first search to check if the connectivity and all of that. I'm so happy that I don't do that anymore. If I want to do it, I just add one line as a plugin, and I just don't have that code sticking out. So I think some of these things were important. I remember in your original paper, you didn't explicitly have the connectivity checking, but I think at lower temperatures you don't have to worry about it, and I think you know better. So um, I think post this thing, there are certain things you did to sort of take care of any breakage of cells or so on. I think so. Yeah. So and I also remember you warning me that the code for basically checking connectivity is doesn't scale very well for three-dimensional system. If so, yeah. So for people who are not sure what I'm talking about, I think if you see plugin, there is something called the connectivity global and connectivity. These are the two plugins I'm talking about. Sometimes you want to make sure that not sometimes, even you always want to make sure that your biological cell is a spatially contiguous cell. Every pixel has a path connecting to the other pixel. And this, at very high energies and so on, if you run the simulation, you might see some cells fragment and so on. Some pixels stay away from the cell and so on. So in this kind of cases, if you're worried about it, I think in CompuCell3D, you can in a very straightforward manner add this, and it doesn't come with, I mean, I haven't quantified it exactly, but it doesn't come with much of a time cost when you do this, at least in two dimensions for sure. So um, one can one can always have this as a safety, and then maybe get rid of it later if they don't feel the need for it. But that's a very handy tool to have. Um, um, I think um, I've used a bunch of these things here for later on in the projects. So I've definitely used the secretion plugins, um, the length constraint, which becomes useful when you don't want to simply when you want to maintain cell shapes that are not just circle in 2D, but you want more elongated cells and so on. Sometimes maybe I don't know, you modeling a myocyte and things like that. If you want your cell to be more spindle and long, then length constraint is a very useful tool to have. So yeah, I think it I think it makes a big difference once you know what can be done. And then I think it's quite easy. Once you know that it can be done in CompuCell3D, I think you'll always find a way to do it. It's just sometimes that was the part that always took me a while to realize, can this be done? I mean, and fortunately for a lot of the times, you have multiple ways to solve it. It's just your task comes down to basically what is the best way to do it in CompuCell3D. Once you do that, I think it's pretty useful. Um, yeah. Yes.
Okay, great. If somebody wanted to explore this simulation and maybe use it to do some simple extension, what would you suggest is something they could take the code that you gave them and add on to it that might be interesting to try?
Yeah. So I think I definitely think something that I didn't talk about, but I think was something that will, I mean, a bit I alluded to multiple times, which is to have multi-compartment. I think um, so. I think if not, you will learn this very soon. Which is that, like right now, we have contact. I mean, to be fair, this code doesn't have contact because I have AdhesionFlex. But just to show you what it does, if I add contact, see, it basically did was every pairwise different cell type, it basically has this contact energy, which I don't need it. It's because I will go with AdhesionFlex because I explicitly model the molecule. But this is how most of the time your model will, this is what you'll be using, I think, in many cases. But just to give a flavor, if you add something called a contact internal, maybe a new line I have never explored. So, for example, you can always try this. Yeah. So when you work with multiple compartments, you will have two different interaction energy terms. One is that um, there is a way you say that this, so multi-compartment cell will be just different cells, but you will say that these such and such cells are now part of the same compartment. And then you can define set of energy interaction energies for things that belong to the same cell or base compartment, I know this thing, and the ones between this. I think this is important because I think, for example, you could have um, a particular cell in the ring. You can say that I will split my edges into like top, bottom, left, and right, and I have a middle which is the and that is the difference. So I can make each cell into a 5 compartment, which is like left, right, top, bottom, and the center. And now every cell has this, and so I can have same properties for the top, bottom, left, and right. But then I need to also show what is the energy between top and left, as long as they belong to the same cell, as opposed to top and left that belong between different cells. So you realize very quickly why you need this contact internal, which is to basically tell there is an interaction energy which is different when you belong to the same cell. And this is very, I mean, it's almost impossible to navigate without this, which is you need to keep track of the fact that interaction is different when you belong to the same cell, and it's very different when it's between cells. So I think that is something very nice to play around with. I think the most basic one to start here, I would say, is to add a nuclei to all of these cells. I think that would be or maybe the simplest, make the germ cell have a nuclei. Forget about an epithelial cell probably can attain them later, but for a germ cell you can have a nuclei and see what it does. I think that would be nice. Um, Another way is then maybe make the static move away from the static field, make the germ cells probably consume it. I know, so there will be some turn on the decay at the site of the germ cells maybe. so that then if you allow the diffusion constant to be non-zero, then you know that there will be net flow towards the region where the germ cell is, because germ cell is the is a sink and it's consuming the field. I think it's a good way to play around with the field. I think it's a useful way to extend. Yeah. So, yeah, I think this is a good starting. I'm not saying that the problem itself is something that you should be like all extending and fascinated by, but I think it's a good template on which you can add these features and see for yourself, because it as I've demonstrated hopefully clearly that it doesn't use many advanced things. It is massively underutilizing the flexible solver. Um, so I think it's a good place to tweak there, and it doesn't use multi-compartment. I think that's an easy adding a nuclei is probably an easy way to do, and I think there are biological contexts where we know this, where I think macrophages in Drosophila, when they're migrating through uh, uh, narrow gaps, nuclei apparently goes to the more anterior part of the cell, on the leading side of the cell, because you want it, because that kind of dictates how much can you squeeze. You can't squeeze beyond whatever nucleus. So there are some interesting properties of this. It would be nice to see if some of these things emerge naturally, or under what constraints can these things emerge. I think that allows for this. I mean, I also training in physics, and so James is a physicist, so I have always tried to not add features that I'm not going to use. It's very, I don't know, it's like a Chekhov's gun view of things. If I have something, I'm going to use it. So, so that's why I kept it very simple. But I think there's no limit to it. So I think if context demands it, I think you should use it, and you can totally do that very easily. Because I can imagine, I never wrote a code where I did a multi-compartment cell in my this thing, but I know it would be a nightmare. Not a nightmare. It would be just very painful to do it. But I think it's much simpler, and you realize just the only trade-off being having an additional contact internal plugin is a very small price you pay for having that feature.
Okay, thank you. Any questions from our audience? Were people able to run the demo? I was impressed. I downloaded it and ran the first time. So worked for me, both of them. Okay, well then I want to thank you very much for coming and talking to us today. People are saying they were able to run it, and the people in the chat are saying they were able to run it. That's great. Look forward to hearing more about your work as you go forward. And thank you for taking the time today. So
thanks a lot. My pleasure. And thanks for hosting this, and thanks for creating with other developers the CompuCell3D, and also, I mean, also the it's not a bad time to thank for Teleport's model as well. So thanks again. Thanks for this opportunity. Take care.